anatomy and electrophysiology. So as I said earlier, electrophysiology is one of these interesting subjects. If you understand the principles of electrophysiology, you will be in a good place for next semester when you learn how neurons work. So cardiac muscle cells exhibit autorhythmicity. So, we've already talked about action potentials, but autorhythmicity is the ability to set their own action potential, essentially. And you have pacemaker cells, which kind of help establish a way for this to be done. Because what they do is they rhythmically and spontaneously generate action potentials. And this spreads to the other types of cardiac muscle cells known as contractile cells. So it's very much like if you've ever been in one of those situations like where one person starts dancing and everybody else catches the fever and starts dancing too. So cardiac myocytes are structurally and physiologically similar to skeletal myocytes. They have the same general function, which is to generate tension, and both generate tension through the sliding filament mechanism of contraction. And cardiomyocytes are branch cells with a single nucleus. They're shorter and wider than skeletal muscle fibers, but as you can see here, many of the structures are structures we're already familiar with. We're talking about a sarcoplastic reticulum, T-tubules, sarcomeres, myofibrils, I-bands, A-bands. These are all things we've already learned about from our study of skeletal muscle. And cardiomyocytes have high energy demands, so that means that they have abundant myoglobin. In fact, mitochondria compose nearly 1/2 of the cytoplasmic volume. Adjacent cardiomyocytes are joined by what we call intercalated discs. So intercalated discs include desmosomes, which hold the cardiac muscle cells together, and gap junctions, which allow the ions to rapidly pass from one cell to another. And so this allows these cells to be, electrically speaking, one large cell, right? So even though they are uninucleate, because of the gap junction, they can function as a set of cells. So cardiac muscle cells also contain selective gated ion channels in the sarcolemma, and the opening and closing of these ion channels is responsible for both the pacemaker and contractile cardiac action potentials. And there are 3 types of gated ion channels. The first one is the voltage-gated sodium ion channels, and they open in response to voltage changes across the membrane, and they're found in all cardiac muscle cells except for certain pacemaker cells. Then we have calcium ions, and they demonstrate voltage-gated opening but time-gated closing, meaning that they close after a certain period regardless of voltage. And all types of cardiac muscle cells have one or more types of potassium ion channels. Some of those are ligand-gated and others are voltage-gated. So pacemaker cells undergo rhythmic, spontaneous depolarizations that lead to action potentials, and these spread quickly through the heart by the cardiac conduction system, which is a group of interconnected pacemaker cells. And action potentials are transmitted from pacemaker cells to contractile cells through intercalated discs, and gap junctions function as electrical synapses. This allows rapid conduction of the electrical signal, and a coordinated unit produces a coordinated heartbeat. Contractile cardiac muscle cells make up about 99% of the cardiac muscle cells. A contractile cardiac muscle cell action potential, you see it go from a charge of minus 85 millivolts to 0 or 20 millivolts. And action potentials are the result of voltage-gated ion channels in the sarcolemma, and because of sodium and potassium ion gradient across the sarcolemma. So when it comes to the sequence of events of a contractile cell action potential, we have a rapid depolarization phase, followed by an initial repolarization phase, followed by a plateau phase and a repolarization phase. So let's take a look at the rapid depolarization phase. In this part, a voltage-gated sodium ion channels activate, and this allows sodium to enter the cell. Remember, these sodiums have a positive charge. This rapidly depolarizes the membrane. It goes from -85 to +20. Then we get to the initial repolarization phase. So the sodium ion channels are inactivated and some potassium ion channels open. Now remember, the potassium ions are positively charged, and the concentration gradient is such that there is more in the cytosol than in the extracellular fluid, so the potassium wants to flow out. So the potassium starts to leak out, and this causes a small initial repolarization. Then we have the plateau phase. So here, calcium ion channels open, and calcium enters as the potassium exits. And this has the effect of prolonging the depolarization. And then we get the repolarization phase, and this is where sodium and calcium channels close, and potassium continues to exit, and this helps to cause the repolarization. So cardiac action potentials resemble skeletal muscle fiber action potentials except for the plateau phase. This helps to slow the heart rate, which provides time for the heart to fill with blood. And without this plateau phase, the resting heart rate would be about 15 times faster than it should be at rest. Then let's talk about the plateau phase. It also increases the strength of the heart's contraction, and the refractory period in cardiac muscle cells is so long, however, that cells cannot maintain a sustained contraction. So the plateau phase, one of the things is that the prolonged action potential makes the muscle twitch last a long time. So it can develop more force, and this allows more calcium ions to enter the cell, and you need this for the cell's contraction via the sliding filament mechanism. Now that refractory period in cardiac muscle cells is so long that the cells can't maintain a sustained contraction. And this allows the heart to relax and the ventricles to refill with blood before the cardiac muscle cells are stimulated to contract again. Then we have the cardiomyocyte excitation contraction coupling, and this occurs by a sliding filament mechanism. Depolarization wave propagates through the sarcolemma. It dives into the cell along the T tubules. It causes the sarcoplastic reticulum to release calcium ions. Those ions bind to troponin, which allows actin and myosin to bind and the cross-bridging cycle to begin. And the cardiomyocyte sarcoplastic reticulum, it doesn't release enough calcium ions to produce a reliably strong contraction. And this is because the SR is actually much less extensive than in a skeletal muscle fiber. So what we need to happen is extracellular calcium needs to diffuse into the cell via the T tubules. And this is where this is why the concentration of calcium ions in the cardiac ECF is so important. So pacemaker cells make up only about 1% of the total of cardiac muscle cells. There are 3 populations of these cells in the heart that are capable of spontaneously generating action potentials, thereby setting the pace of the heart. These 3 cell populations are collectively called the cardiac conduction system. So depolarization in a pacemaker cell occurs much more slowly. And that's due in part to the lack of voltage-gated sodium ion channels in the pacemaker sarcolemma. And pacemaker cell action potentials lack a plateau phase, and the membrane potential oscillates. That is, it never remains at a resting level, and instead it occurs in a cycle, with the last event triggering the first. And this is because of something called a non-specific cation channel that is unique to these pacemaker cells. And if we look at that name, non-specific means it's not picking one species, in this case species referring to an ion species, right? So it's not picking calcium ions, sodium ions, potassium ions. It's not discriminating. And cations refers to a positively charged ion, and these are unique to the pacemaker cells. So the pacemaker potential is much different than that of a contractile cell. It has 4 phases. We have a slow initial depolarization, followed by a full depolarization, followed by a repolarization, followed by a minimum potential phase. So let's take a look. So for our slow initial depolarization phase, the pacemaker potential starts with the plasma membrane in a hyperpolarized state. At this minimum membrane potential, and it opens non-specific cation channels in the membrane. This allows sodium ions to leak into the cell and potassium ions to leak out, and this results in an overall slow depolarization to threshold. Remember, because the potassium ions are positively charged, as are the sodium ions. So some potassiums are leaving, some sodiums are coming in. So there is a net change, but it isn't as big as if we allowed one versus the other. Then we get to the full depolarization phase. So when the membrane reaches some threshold, which in this case is minus 40 millivolts, voltage-gated calcium ion channels open, which allows calcium ions to enter the cell and thereby causes the membrane to fully depolarize. Then we get to the repolarization phase. And remember that calcium ion channels are time-gated for closing. So after a certain time, which is about usually 100 to 150 milliseconds, they close. At the same time, voltage-gated potassium ion channels begin to open, and this allows potassium ions to exit the cell, and the membrane begins to repolarize. And then we get to the minimal potential phase. So potassium ion channels remain open until the membrane reaches its minimum potential. When this happens, the membrane is hyperpolarized, which opens the non-specific cation channels, and the cycle begins again. So the cardiac conduction system includes 3 populations of pacemaker cells. So we have the SA node generates an action potential which spreads to the atrial cells into the AV node. And after the AV node delay, the action potential is conducted to the AV bundle and then to the right and left bundle branches. And then the action potential spreads from the bundle branches along the Purkinje fibers to the contractile cells of the ventricles. And the SA node is a pacemaker of the healthy heart, and the SA node rhythms are known as sinus rhythms. An AV node and Purkinje fiber system normally only conduct action potentials that's generated by the SA node. If the SA node ceases to function, the AV node can successfully pace the heart, albeit somewhat slowly. Impulses are then conducted by specialized atrial conducting fibers to the AV node, a process that altogether requires about 0.03 seconds. And once the impulse reaches the AV node, conduction slows considerably as a result of 2 factors. First, there's a low number of gap junctions between the AV node cells, and second, there's a presence of non-conducting fibroskeleton that surrounds that AV node. This slow conduction, known as the AV node delay, generally lasts about 0.13 seconds, and this allows the atria to depolarize and contract before the ventricles, and this gives the ventricles time to fill with blood. So under normal conditions, the SA node generates an action potential. This spreads rapidly via gap junctions to the surrounding atrial cells. The AV node delay also helps to prevent current from flowing backward from the AV bundle into the AV node and atria. Action potentials are then conducted from the AV bundle to the right and left bundle branches, and depolarization spreads along the Purkinje fibers to contractile cardiac muscle cells of the ventricles.